Welcome to Biotech Stage, a stage where we talk with biotech professionals from around the world and also from the whole spectrum of job functions, including presidents, vice presidents, entrepreneurs, CEOs, directors, team leaders, and most importantly, the people who work on the bench. That means scientists. So, welcome again. Today, we welcome Dr. Ivan Bruno as our guest. Dr. Ivan Bruno has been in the industry or biotech industry or cell and gene therapy industry for over 2 decades. She started her work at Gene Medicine, a company based in Texas, and then she grew to more senior positions and she oversaw the preclinical work at a company named Cytoimmune. So, welcome, Dr. Bruno.
Thank you. Thank you for having me. I'm really happy to be here and I appreciate the invitation.
Dr. Bruno, there are a lot of things to be learned from you. There are a lot of things to be discussed with you. But one thing that comes first in my mind is that you were working as a scientist in a gene therapy company, and then you are working as a vice president of preclinical affairs. So, how did you handle such a big change? Because the responsibilities for scientists are very different from the vice president level position, especially in a field of gene therapy where things are changing so fast. So, could you please say something on that?
Sure, at the scientist level, you are more involved hands on with the aspects of experimental design, experimental execution, data analysis. That never leaves your role. So, from being a scientist to being a VP, always I had to keep in mind that my role was science driven.
Mhm.
And it's very important that science is always the center of the decisions. Now, once you're in a more senior position, that's when you incorporate not only the science, but you incorporate regulatory, clinical. So you see it all in a much broader way, and you incorporate all of that into your role. But I always think that once a scientist, you're always a scientist.
Yeah, that's true. And one other thing which I wanted to find out from you is that industry has changed a lot in past 15, 20 years and definitely will keep on changing. So how do you see, I mean, the changes are happening and where do you think industry will be in coming 10 years, 5 years?
So if you go back and you think about the industry, 2 decades ago was basically dominated by monoclonal antibodies, small molecules. Now we have platforms like viral vectors, RNA, cell therapy that are really creating an impact in the development of therapeutics that are more and more effective for some of these indications, oncological indications, autoimmune diseases. So we're seeing the applicability of all of these therapies, especially in cell therapy. Now what I see happening is basically is moving from the strenuous manufacturing and external ex vivo expansion of these cells into more in vivo. So now we know these therapies work. The question now is how can we make them more affordable? How can we make them more efficient in a way of the way we manufacture, the way we distribute, the way we make them commercial? So that's we're seeing an increased awareness of in vivo approaches, and I think that's something that the field is gonna welcome and is gonna expand. Then we also have the intelligence, and
Mhm.
how is that being incorporated into manufacturing, into design, into even clinical design or clinical data analysis. So all of that is going to be incorporated into what we're seeing now and what we're about to see in the near future.
Oh, okay, okay. Yeah, you touched based on AI, and that's what I wanted to ask you that when it comes to change, machine learning and artificial intelligence is making a big difference. And I understand that you could took some courses also on artificial intelligence and ML. So, do you want to say something on that?
Yes, actually, I was fortunate to take a course, an MIT course in the applicability of AI in biotech. And it was eye opening. We know how AI is really revolutioning the field of discovery and how now molecules can be defined, they could be predicted, and their function can be predicted with AI technology. But beyond that, I mean, I even saw how you can see a cell in a pre-cancer stage and a post-cancer stage, and AI can pretty much predict that transition, how that transition will happen. So it can give a the researchers, for instance, it can give them an opportunity to have diagnostics information. So how would it look in that in between stage? Also, even for clinical, analyzing clinical data. So clinical data can be overwhelming when it comes to evaluating the patients, and all that information sometimes could be for a researcher, for the clinical researcher, can be a challenge. And how AI is becoming a more and more a tool to capture that information and make it meaningful, and helping decipher what it means, and how that relates to disease or therapeutic benefit. Same with manufacturing, aspects of manufacturing technology, the use of bioreactors, understanding the behavior of the cell during the manufacturing, the potency, the cell morphology, how the cell is behaving, the cell
Mhm.
characteristics, the phenotype.
Mhm.
All of that is now information that you can clearly capture and analyze and put it all together. Not to speak about genetics, I mean, understanding what is changing at the molecular level, what is changing at the genetics, that what is a genetic change is making, what is happening to the cell when you make these changes, and the tumor microenvironment, which we know is highly complex. So all of that is now information that can be captured and processed with the help of artificial intelligence, and it is here to stay. That's the reality.
Ah, okay, good to know. So, the next thing that I wanted to discuss with you is that in any field, the continuous research is obviously needed, new products, new research. And when it comes to biotechnology, the new research obviously means new research can only be successful if there is a sufficient money. So, that means the funding. So, I just wanted to get some of your ideas on how, I mean, what is the way to get the new funding or how the industry is shaping up for that. So, if you could say something on that.
Yes, and that's a very important question. I mean, almost every single role that I've been, it's been very important to understand how we can bring in more funds,
Mhm.
where to look for these funds, and what is a fundable program. So it's this is a in reality this is a multi-phase, a very strategic process. You need to understand the scientific value
Mhm.
of your product. You need to understand the feasibility, and when it comes to feasibility, the manufacturability, the regulatory feasibility, even the ethical feasibility. Yeah, it does it meet all these requirements? And then take it even. It's never too early to think about the commercial potential of that. What are some of the gaps that you're feeling? Why is your product standing out from everything that is already existing? So those are, you need to think strategically on how your product can fill all these different gaps. And then you go, after you strategically think of all these, the value of what you're developing, you
Mhm.
look and see which sources of funding. So there's non-diluting funding, such
Mhm.
as grants. There's the NIH-based grants. For a startup, for instance, it could be if you are transferring a technology from an academic institution, there's what they call STTRs. There's also SBIRs that they may cover different stages of development from preclinical, from discovery to preclinical to clinical. And but they will ask you about a commercialization plan. How would you take this into a commercially viable product? Then there's venture capitalists, and a little bit harder these days. You really have to demonstrate the feasibility. So what is the data that you will need
Mhm.
to provide evidence of the value of what you're developing? Think about what in vitro studies, what in vivo studies you will need in
Mhm.
order to provide evidence of the value being proposed. And then there's other sources of funding that could also be available to you. You can look into partnerships, for instance.
Okay.
Partnerships are always a very good idea. One does not have to have it all your capabilities. So be strategic about who you partner and why you partner and how that partnership will be formed, and roles and responsibilities. And in many of these, we're seeing it more and more. We see mergers, we see acquisitions, we see partnerships where you can see the value of the combination of the capabilities of these different entities. So that's something very important to consider when looking at funding.
Okay, thank you very much. The people need to think not just about the basic research, but also they need to keep those 3 points that you mentioned: scientific validity and feasibility, not just the, I mean, overall feasibility, including regulatory and ethical. And third, the commercialability. So basic research on paper, yeah, science is good, but these 3 points also they need to focus a lot in order to get the funding.
I've seen in a lot of the technology transfers, for instance, when I was working at a CDMO,
Mm-hmm.
a lot of these technologies have potential, but they're very early
Mm-hmm.
and they have not taken into consideration the regulatory aspects
Mm-hmm.
of the product.
Mm-hmm.
So that means it's manufactured as in a bench. What you manufacture in a bench does not necessarily can be manufactured as a drug product.
Yeah.
So these are things to consider, and you don't want to invest a lot at the beginning until you understand that indeed you will take this product into a drug development pipeline, but consider having the thoughts early on of what could be a way of transitioning, of translating these products. And that's when consultations, having people with the expertise in drug development
Mm-hmm.
early on, being involved and being part of this early stage of development, it's very important.
That's good to know. One other thing that I wanted to ask, especially since you are expert on the cell and gene therapy, and that is a lot of people have, especially after the COVID era, people are more aware of biotechnology or cell and gene therapy, and sometimes when they are aware, that doesn't mean that they are aware on the good things. They are aware on some of the negative perception. So, what do you think the public perception is on the biotech or some of the techniques that are used in biotechnology?
I think that's a very interesting question, and I actually have experienced the positive and the negative.
Hmm.
In the positive, for instance, a good example we see for COVID, we saw both positive and negative.
Hmm.
Positive, we saw the effect of science, of research can have in something as critical as a pandemic. I mean, we were able to identify a sequence quickly, that protein, and then use it as a vaccine. And because there was already know-hows on how to manufacture RNA, it was very easy. There were aspects of it that were challenging, and upscaling, for instance, formulating it at a very large upscaled amounts. Yes, that was mainly a challenge. But I think the public was able to appreciate how fast that was
Hmm.
done and the value that it had in the public, for the public. The economic impact, so job creations, investments, scientific value of what is being developed. So I've seen the public appreciating a
Hmm.
lot of these aspects of science. But I've also seen the negative, and between the negative, you'll see, for example, there was a lot of misinformation. So perhaps the scientific community could have done a better job explaining. I don't know. There was a lot of one thing that I thought was really interesting. It was the fact that they were saying that from the vaccine, the COVID vaccine, the RNA will go to different parts of the body,
Hmm.
and I was thinking, wow, I had a very hard time keeping RNA from degrading.
Yeah, RNA, once it goes into the cytoplasm, it has a very short shelf life.
Public doesn't know this, and there was a lot of misconception. There's fear of the unknown. RNA was unknown, For us molecular biologists, RNA has been around forever. But for the general public, it was an unknown. It wasn't. There's a lot worse vaccines that are highly effective, and they're not afraid of them. But something unknown could create fear in the public. So, I think these were symptoms of there was a communication gap. Perhaps using nomenclatures, using scientific jargon that was more in lay terms. Perhaps making minimizing the misinformation, making sure it was addressed. I mean, there may be ways to address that, but also in another aspect of the public, the importance of the public. I've seen in, for instance, having a clinical study when you're enrolling, having your advocacy groups.
Hmm.
I've had a very positive experience with advocacy groups because they understand the needs of
Hmm.
the public, of the patients, and then they create a bridge between the drug developers and the community. So I think they play a very important role, and I believe that the scientific community should be fully engaged with the public, and especially through advocacy groups to
Okay.
drive the science.
Okay. I think that that's important point. The advocacy group trying to educate public and the clinical, clinical and the biotechnology terms and aspects. Okay, Dr. Bruno, I wanted to also ask, since you have so much of experience, what is your advice for new younger generation who are entering into the biotechnology, who has done the bachelor's or master's and have worked on bench for 2 3 years, and maybe they are in maps? Should they move to cell and gene therapy? Because cell and gene therapy is advancing, CAR T cells everywhere, stem cell research everywhere. So, what is your advice on that?
The first advice that I always give to young scientists is choose something that you're passionate about, because
Okay.
the field can be challenging, and more often you will see that you will fail at something, but that doesn't mean you fail. It means that you obtain information that could help you define the next steps.
Mm-hmm.
So being passionate about what you do, grow, learn.
Mm-hmm.
Don't limit yourself. One thing that I've in my career I have seen it has helped me is that I don't limit myself on I'm only process development, I'm only analytical, I'm only.
Mm-hmm.
So you will see me working closely with the manufacturing team, with the
Mm-hmm.
quality assurance team, with the quality control teams,
Mm-hmm.
with the regulatory team.
Mm-hmm.
So wrap yourself in all of these different aspects of drug development that are critical. They're all a
Mm-hmm.
piece of the pie, right? And learning, adapting the new technologies. And I've worked with young scientists that you give them some information and they run with it and they
Mm-hmm.
take it to the next level. And they
Mm-hmm.
come back with a massive amount of information, and that's something that I totally appreciate. And you feel that you want to take that person to the next level. So
Mm-hmm.
it's very important to be very critical, to
Mm-hmm.
be very passionate, and to be hard worker. So
Mm-hmm.
go beyond what you're called to do, in a
Mm-hmm.
sense of always try to grow and always try to see what the next step is.
Okay, okay. These are very important points. And do you think you can also suggest some of the obstacles, some of the things they need to watch out for? They need to be more careful.
Yeah, it's so biotech in general, it's a very volatile
Mm-hmm.
industry.
Mm-hmm.
I've seen it myself. I've been
Mm-hmm.
part of several startups. And some make it, some don't. And it just never take that personal. I mean, every experience is an opportunity to grow.
Mm-hmm.
And good or not so good experiences are always opportunities to grow. And I believe that that's something that you should always keep in mind.
Mm-hmm.
And it has nothing to do with you personally, it has to do with the industry.
Mm-hmm.
And just seek what's your next challenge.
Okay, okay. Yeah, okay. That's good to know. And one question actually I forgot to ask, and I should have asked you in the beginning. And that is what is your opinion on the cost of the final drug product, final biotech product?
Yeah.
Because I mean, something could be very, very expensive. So, do you think it is a cost of the raw materials that will bring the cost down, or do you think it is a overall process efficiency, or something else? So, I wanted to get your feedback on that.
Yes, actually, I've seen the cost is forbidden for the average individual. For instance, the cost of cell therapy is so high.
Hmm.
I've seen patients struggling to get the current approved cell therapies because of they have engaged in a battle with insurance.
Hmm.
So it's highly expensive.
Hmm.
I think this is related to several things, and I think it's in it could be raw materials, it could be the cost of labor, it could be the complexity of these drugs, making these drugs. For instance, if you're making a cell therapy that is genetically modified with viruses, the virus is a separate drug product that you have to manufacture GMP in order to generate a clinical grade material.
Hmm.
So all of that adds up to the cost. But
Hmm.
one thing I want to mention is at the Biostage conference a couple years ago, I had just had an approved a product, and it was just a fraction of the cost of what you see here in the US.
Hmm.
So there's something to learn there. We probably
Hmm.
need to understand how we can make this product in a more cost effective. Maybe
Hmm.
we can work with regulatory agencies, with the federal agencies, with the FDA, EMA. Maybe these clinical studies could be reduced or accelerated, or some of these requirements could be, once they're established, they could be minimized.
Okay.
So there's several aspects of drug development that I think will need a closer look to see how we can reduce the cost. The ex vivo expansion of the cells as of now is very expensive. How the use of, for instance, bioreactors, allogeneic therapies that may be more cost effective, or
Okay.
maybe the autologous that could be done in a less expensive manner. So there's a lot of things that we can look into, and I think the field is moving in that direction.
Okay, that's good to know. So, Dr. Bruno, with this, I think we are now coming towards the end of the this conversation or podcast. So, for the end, I wanted to have in two sections actually. In one first section, I'm going to ask you or request you to share something funny, something surprising that you might have noticed in your career, and or you could share something that you think exists in biotechnology or cell and gene therapy, and public is not aware of about it. And the second part I will do by a rapid fire questions where I will ask you 10 different questions, and you just need to respond in on a scale of 1 to 10. So can you please share something funny or exciting fact about biotechnology?
Okay, well, something funny. I've had several funny events, and one of them has been with one of my younger scientists.
Hmm.
One time we were at a meeting, and here I am thinking in a very complex way how to solve an issue that we were talking about. And the young scientists very candidly say, "Well, why would we don't do this?"
Hmm.
And it was a very simple, very straightforward solution. Guess what? That was the solution. So I've always, ever since, I've always have all I take into consideration from the most junior to the most senior, their opinion, their look. I ask them to critically think
uh,
and criticize what I'm proposing. So I give them that opportunity to be part of the solution. And it's amazing what you can do when you do that. I've also, once I had a co-worker, I was in the manufacturing team with them. That was actually recently with Cytomune. And we worked together for several weeks. And when she saw the sign on my door and she said, "You're the VP?" And she got scared. She said, "Well, you were there with me." And I'm like, "Yeah, well, that's okay. I mean, I want to be part of the team." And she was amazed that I was there with them, working in the clean room, manufacturing and looking and see how the product was being manufactured. So this is it's just fun occasions where I see the value of taking working together as a team.
so nice of you to to be in the clean room and work with the manufacturing team. And I think the thing which you mentioned about the young scientist idea, I think that's a big encouragement for all the people who are entering into the science, that they don't need to be they don't need to be hesitant in saying something. They
Exactly.
may be having the world's biggest idea, and even if that they don't have the world's biggest idea, sometimes their feedback is could be very, very important. So
Very valuable.
they don't need to be shy. They need to speak up.
Yes, I totally agree. And I try to encourage that all the time so that they become part of the solution. They grow very fast when they feel comfortable
Okay.
sharing their thoughts and being critical as well.
Okay. So now I'm going to start the rapid fire questions. So in this rapid fire questions, there I will ask you 10 different questions, and as I mentioned, you need to answer from a scale of 1 to 10, and 10 being the best or almost there or strongly agree, depending upon the type of
Okay.
question. And if you're not sure about any question, because most of these questions are related with India, just say not sure. So I'll start right
Okay.
now. Okay. So question first. Quality of biotech drug products in India.
9,
Production capacity in India.
10,
Okay. Innovation in India.
4,
IP protection in India.
3,
Okay. Adequate availability of talented workforce.
10,
Okay. Productive interactions among peers.
10.
Okay. And capital available, public or private, to support commercialize new products.
Not clear.
Okay. Cost efficiency with the aim to reduce the cost of drugs.
10.
10. Okay. Question number 9. Unambiguous guidelines or regulatory process.
Not clear.
Okay. And finally, the question number 10. Make in India efforts.
10.
10. Okay. Okay. So that ends our rapid fire questions. So, Dr. Bruno, with this, I think we had very nice conversation with you, or this podcast about the biotechnology and cell and gene therapy and your all experience. And I'm so glad that you talked about the machine learning, artificial intelligence, and especially the advice and coaching that you gave. And I mean, overall, the different ideas that you gave, I think that will be very valuable for the new generation working on the bench. So thank you very much.
You. Thank you for the invitation. I totally enjoyed it, and I look forward to seeing more and more feedback from KOLs in the field. So thanks a lot.
Thank you. Thank you.